This video is a learning aid to help intro to biology students get a grip on meiosis. Meiosis is the process by which unique gamete cells are formed, each having half the DNA of the original parent cell. Meiosis starts by the pairing up of homologs. Homologs can be identified by the fact they have the same gene on the chromosomes. For example, right here, chromosomes B, this chromosome and this chromosome are homologs because they both have B and B in the same location. These homologs will pair up and come to the middle. Same situation for this one. This chromosome is R, and this chromosome is also R, so these will line up. However, homologs don't always behave like that. This is reserved for autosomal cells. For sex chromosomes, you instead get this situation, where here you get a X chromosome with the gene on it, and here you get a Y chromosome that is lacking the gene. However, since these are X and Y, they operate like homologs regardless. The next step in the process is replication. In replication, the gene should stay the same on the chromosome. For example, if we look here, this is a big B chromosome. But when we open it, we see that we replicated it. There's still one chromosome, and each gene on the chromosome is identical. So unless recombination happens, you should only have two big B's on this chromosome. If we go to its homolog, a similar thing happens. Two little B's. Now let's replicate the other chromosomes. Now that we have replicated all the chromosomes, you can see here, this chromosome is one chromosome with two sisters that are identical. Same here and same here. When you replicate a chromosome, unless there's recombination, your genes should remain exactly the same on each chromosome. There should be a little b here, little b here. Big R here, big R here. Now, we can begin meiosis 1, where we separate the homologs. And they enter their own cells. That is meiosis 1. Now, let's begin meiosis 2. We're going to focus on only one of the newly formed daughter cells. So we're going to move this over here. In meiosis 2, we simply do a similar process, where we split it in half one more time. So we're going to cut it in half again. As you can see here, we separate the sister chromatids. We separated them. Since we have separated sister chromatids, they are now completely new chromosomes. And you see here, we end up with half the DNA. Usually in processes such as meiosis, we would end up with the same amount of DNA as the original cell. But now we only have half the DNA as the original cell, as they don't have any homologs to go with them. And that is meiosis. Now, that process is good if you're trying to understand how to do meiosis. But if all you need to do is answer the question and get the test done, follow this process. Now, as you can tell by my large hands, these stick figures are not me. However, they represent my emotions. And this is me begging and pleading on my hands and knees. Please, I need you to pull out your pencil. And when you do a meiosis, anything, mitosis, meiosis, genes, gametes, pedigrees, please bring out your pencil and draw for me. I promise you, the difference between a C student and a not C student is pulling out the pencil and just drawing. I promise you, it makes your life 10 times easier. And it also makes me 10 times happier. Now, in order to do the process, it's quite simple actually. What you want to do is you want to draw your homologs. This is where you start. Then, you're going to want to draw them after you have replicated. Once they have replicated, all you have to do is draw it 2 more times. That's okay. Now, right here, this will be meiosis 1. Right here will be meiosis 2. And now, it's gonna get real simple. Starting at meiosis 1, what you're gonna do is draw a line starting at meiosis 1 and going all the way down. Then, at meiosis 2, all you gotta do is draw another line in the middle of that, another line in the middle of that. And just like that, you have done it. You now have your cells. Just to show you guys how this works. If I were to draw them and I were to draw genes on them, I could replicate it. Remember, when you replicate it, the genes on the chromosomes should stay exactly the same. Now, all you gotta do is draw a line here, draw a line here, and draw a line here. Focus on this area right here. And what we have is B, 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 B. And that way, you have drawn the entire process for meiosis. You see that? It's that simple. I hope this video was able to help you guys, and I hope you guys are able to succeed and progress through your university journey. Thank you, and have a good day.